Hello there. In this video, I am going to go over the process of transcription and translation and protein synthesis. At the end, I'm going to talk about what CRISPR Cas9 is. And again, this is a very basic video. I just want you to get an understanding of what these processes are. Because we talk about proteins throughout the whole semester, I want you to have a general idea of how those are made and why they're made the way they are. Okay, so here we are in the nucleus with our double strand of DNA. Remember, within this strand of DNA are areas, genes, that are going to give us the instructions for the proteins of our body. So think of the strand of DNA, the gene, the area that's going to give us the instructions for the protein as blueprints. Okay, they're the blueprints for the different proteins of your body. Imagine you made some product that was going to make you millions of dollars, and you had that blueprint. You would not want that blueprint to leave the protected area, whether that's in an office, in a safe, or whatever. I want you to think about the nucleus as being that protected safe or office in your factory, and you want it to stay there. The body does the same thing. So let's pretend we're making insulin, which helps regulate and maintain homeostasis of your blood sugar. So let's say for breakfast you have ice cream and Fruit Loops and a bunch of sugary stuff. Your blood sugar will start to go up. The sensors will sense that your blood sugar is going up, and it's going to want to bring it down. And so in your pancreas cells, it's going to activate the process of making insulin. So let's imagine that this gene right here, this red strand of DNA, that is the instructions for insulin. We don't want that strand, the original blueprint, to leave the nucleus. So what's going to happen is there's a molecule called mRNA, and mRNA is going to make a copy of that gene. So we're going to make a copy of that gene right here. Okay, that's the blue strand here. This process of going from DNA, or the gene, to mRNA is called transcription, and it occurs in the nucleus. So DNA to mRNA is called transcription, and it occurs in the nucleus. Then what's going to happen? Again, the gene is not going to leave the nucleus. The strand, this copy of the gene, is. So it's going to leave the nucleus, and it's going to head to the ribosomes of the cell. Okay. What's going to happen now is, based on the order of these nucleotides, we're going to put together a protein using a molecule called tRNA, or transfer RNA. Now, I like this drawing except for one part. The part I don't like is that each of these squares is representing an amino acid, and you wouldn't have just one amino acid being made here. So remember, there are 20 different amino acids in your body. Imagine each of them represented by a different color. Based on what is on the mRNA will determine which tRNA will bind there and which amino acid will be on the other end. So imagine you've had this combination of colors that are going to end up making insulin. So we get a tRNA with a amino acid on the one side. They are then going to bond to each other in what's called a peptide bond, and we get then a protein, also known as the polypeptide chain. So in our case, this molecule would be insulin. This process of going from mRNA to tRNA to a protein is called translation. And it occurs at the ribosomes of the cell. Now you have this insulin, which, if you remember your cell parts, then is most likely going to go into the endoplasmic reticulum, where it's going to be shaped, checked, and then shipped, go to the Golgi apparatus, where it's going to be modified again, and then shipped. In this case, it would be shipped out of the cell by exocytosis into the bloodstream, where it will then bind to receptors to help control blood glucose levels. So again, this process of going from mRNA to tRNA to the protein is called translation, and it occurs at the ribosome. So what we've done is we've gone from the gene here, made a copy with mRNA, transcription, in the nucleus. That mRNA strand has left the nucleus, going to the ribosome. We've then gone from the mRNA to tRNA, each which have different amino acids attached to them, Based on these instructions, we get a protein. We get a protein, in this case insulin. Okay, in this case insulin. Also, the order of amino acids in this protein are then going to determine the shape of the protein. Okay, so the shape of the protein is going to be very important. In the case of insulin, remember it has to bind to an insulin receptor. So it has to fit. It has to fit in that. And so that's what's going to happen. Okay, the order of amino acids is going to determine the shape of the protein. Okay, so what happens? What is CRISPR Cas9? CRISPR Cas9 is a relatively new technology that is going to allow us possibly to modify the gene. So I'm going to take a disease like, let's use cystic fibrosis, where they do not make channel proteins for chloride. So the channel protein, which you've talked about, the channel protein for chloride in people with cystic fibrosis does not function. So chloride cannot move in and out. Why is that? It is that way because they have a mutation in the gene. These nucleotides are not correct. The gene has a mutation, which they are born with. So what happens is they still have the gene, but the copy of mRNA is not correct because the gene's not correct, and so the protein that ends up being made is not correct. Now, we don't have a cure for cystic fibrosis. We can control the symptoms of it. So the lifespan of people with cystic fibrosis has increased over the years, but we don't have a cure. What would be the ultimate cure to a genetic disorder like cystic fibrosis? The ultimate cure would be to replace this gene. So if we could cut out the bad gene, so imagine taking scissors, cutting out the bad gene, and then replacing the bad gene with a healthy gene. Now you've got a healthy gene. Now you end up with the proper protein. This is what CRISPR-Cas9 has the ability to do, to cut out a bad gene and put in a healthy gene. Now obviously it's much more complicated than that, but that is the direction that CRISPR-Cas9 research is going. So I just want to introduce you to that. There's some controversy to it. Yeah, you can change a person from having a disease to not having a disease, but does that give us the right to, well, if we want to make a person taller, can we change a gene that will affect their height? Right? Can we change eye color? Can we make them a better athlete? So by changing genes, you can be curing disease, but you can also be modifying the person. So that's one of the controversies dealing with CRISPR-Cas9, but I just wanted to introduce it to you as a technology. The last thing I want to do in this video is look at genomic medicine. I know this is hard to read. I'm not expecting you to be able to read it, but these are all of the chromosomes, and all these different circles here with these different colors are a map of the genes and what they can affect in a person's body. So let's say a person has high blood pressure. If you went through all of these, which again, I don't expect you to do, you would note that there are several genes amongst these chromosomes that are linked to high blood pressure. So genomic medicine is that in the future, based on your genes, what genes you have, what mutations, what differences you have in those genes, may help determine which medication is going to help you. So most people that have high blood pressure, it's what's called idiopathic. There is no known cause. It has a genetic component to it. So if in the future they can figure out, well, it's because of this mutation and this mutation that you have high blood pressure, then this is the drug that's going to help you. Now, if you have high blood pressure and it's different genes that are affecting why you have high blood pressure, it may be a different medication that's going to help you. So maybe someday that you will walk into a doctor's office with a little container or a little disc that has your genome in it, okay, with all your genes and mutations. The doctor then knows that this medication will work for whatever problem you are having. They also use genomic medicine for cancer treatments. So if you have cancer cells and they can do a DNA study of that and they see that you have certain mutations that have caused that cancer, there may be a specific drug that will help you. If you have different mutations, then they know that these drugs won't help you, but maybe this one will. So that's where medicine is going. It's going to be a while till we can do that. With some cancer treatments, though, they can already do that. They can determine that you have these mutations, we'll give you this drug, and there's a very good chance that it will work. Okay, so that's just the basics of some genetics that I want you to understand. If you have any questions, please let me or your instructor know. Thank you.